Hi everyone, I'm Madeline and I'm going to be going over the regulation of the gastric phase of digestion. Once food enters the stomach, stimuli in the gastric lumen initiates a series of short reflexes that make up the gastric phase of digestion. G cells, which are found deep inside the gastric glands, secrete this hormone gastrin into the blood. And in these short reflexes, gastrin release is stimulated by the presence of amino acids and peptides in the stomach and by distension of the stomach. Gastrin release is also triggered by neural reflexes. So short reflexes are mediated by the enteric nervous system neurotransmitter called gastrin releasing peptide, or GRP. In cephalic reflexes, parasympathetic neurons from the vagus nerve stimulate G cells to release gastrin into the blood. Gastrin's primary action is to promote acid release, which I'll go into greater detail about in a little bit. But it does this directly by acting on parietal cells and indirectly by stimulating histamine release. So the lumen of the stomach is lined with mucus-producing epithelium punctuated by the openings of these gastric pits. These pits lead to gastric glands deep within the mucosal layer, and multiple cell types within the glands produce gastric acid, enzymes, hormones, and paracrine molecules. So the various secretions of gastric mucosa cells, their stimuli for release, and their functions are summarized in the figure or table seen on this slide. So the mucus surface cells are releasing mucus and bicarbonate, which form a physical barrier and a buffer for gastric acid to prevent damage to the epithelium. The parietal cells release gastric acid, which kills bacteria by releasing pepsin, an intrinsic factor which helps permit absorption. The enterochromaffin-like cell secretes histamine, which stimulates further gastric acid secretion. And chief cells secrete pepsin and gastric lipase, which aid in protein and fat digestion. And then somatostatin is released by D cells, which inhibits gastric acid secretion. And the G cells secrete gastrin, which, like histamine, stimulates gastric acid secretion as well. I'll go into further detail about a lot of these in some of the next slides. So parietal cells, which are deep within the gastric glands, secrete gastric acid into the lumen of the stomach. In fact, acid secretion in the stomach is about 1 to 3 liters per day and can create a luminal pH as low as 1, whereas the cytoplasmic pH of the parietal cells is about 7.2. So what this means is the cells are pumping hydrogen against a gradient that can be 1.5 million times more concentrated in the lumen. So the parietal cell pathway for acid secretion is seen in the figure at the right on this slide. The process begins when hydrogen from water inside the parietal cell is pumped into the stomach lumen by the hydrogen potassium ATPase or pump in exchange for potassium entering the cell. Chloride then follows the electrical gradient, which is created by hydrogen moving through these open chloride channels. And then the final result of this is secretion of hydrochloride by the cell. So through learning and understanding of the cellular mechanism of these parietal cell acid secretion, it's allowed scientists to develop a new class of drugs to treat oversecretion of gastric acid. So these drugs are known as proton pump inhibitors, or PPIs, or more commonly known to us as Prilosec, and some of the alternatives for Prilosec, which can be bought over the counter. They work by blocking activity of this hydrogen potassium pump. So there are multiple gastric acid functions. So acid in the stomach lumen causes release and activation of pepsin, which is an enzyme that digests proteins. It triggers somatostatin release from the D cells, and it denatures proteins by breaking disulfide and hydrogen bonds that hold the protein within its tertiary structure. So unfolding these protein chains make the peptide bonds between the amino acids more accessible for digestion from pepsin. Gastric acid also helps kill bacteria and other ingested microorganisms within our stomach, and it inactivates salivary amylase in stopping carbohydrate digestion, which begins in the mouth. So enzyme secretions. There are 2 enzymes that are produced in the gastric phase of digestion, pepsin and gastric lipase. Pepsin, as I've already discussed somewhat, has the vital function of carrying out the initial digestion of proteins. It's particularly effective on collagen and therefore plays a very important role in helping us digest meats. Pepsin is secreted as the inactive enzyme pepsinogen by chief cells in the gastric glands. Acid stimulates pepsinogen release from these chief cells through a short reflex, as I kind of discussed a little bit, which is mediated in the enteric nervous system, which originates when we taste, see, smell, or even just think of food. As this initiates gastric secretion of gastrin, histamine, and acid. Gastrin stimulates acid secretion by direct action on the parietal cells or indirect action through histamine. And then acid then stimulates the short reflex secretion of pepsinogen. So once in the stomach lumen, pepsinogen is cleaved to active pepsin by the action of hydrogen, and from there, protein digestion can begin. The gastric lipase is co-secreted with pepsin as well, and the lipases are enzymes that break down triglycerides. However, about less than 1/3 of fat digestion actually takes place in the stomach. So paracrine secretions from the gastric mucosa include histamine, somatostatin, and intrinsic factor. Histamine is a paracrine signal secreted by the ECL cells in response to gastrin or acetylcholine stimulation. It diffuses to its target, which is the parietal cells, and stimulates acid secretion by combining with H2 receptors on the parietal cells. These H2 receptor antagonists, such as cimetidine and ranitidine, block histamine action and are a second class of drugs which can be used to additionally treat acid hypersecretion of the stomach. Intrinsic factor is a protein secreted by the same gastric parietal cells that secrete acid. In the lumen of the stomach, intrinsic factor complexes with vitamin B12, which is a step that we need for vitamin absorption within the intestine. The final paracrine secretion, which I'll discuss, is somatostatin, also known as the hypothalamic growth hormone inhibiting hormone. It is secreted by D cells in the stomach, and somatostatin is the primary negative feedback signal for the gastric phase of secretion, making it a very important part of this. It shuts down acid secretion both directly and indirectly by decreasing gastrin and histamine secretion. Somatostatin also inhibits pepsinogen secretion additionally. So if you wanted to test yourself on all the things we just discussed here, there's a little bit of a worksheet about everything I just went over and discussed. And that is it.